Nice to see folks here, and thanks for sharing some interest in the work. So today I'll be sharing, as Johan mentioned, our work on HyenaDNA, a genomic sequence model that we recently, or last year, put out, and we're really excited in extending the work. We're at the cusp of actually a new iteration of the project that's going to come out next month, most likely. But so hopefully we'll tease a little bit about that at the end. But for now, we'll talk about the initial work and why we're so excited about it. For some context, myself, I'm a fourth-year PhD student at Stanford, as Johan mentioned. We're a part of this research group in computer science called Hazy Research, led by Chris Ray. And there's a theme currently where we focus on better foundation models or large language models. And these have looked like a couple of things. One is faster, more efficient versions of these models that can fit longer context, but also these models that we're calling alternative architectures. So these are architectures beyond transformers that could perhaps open up new capabilities that might have been constrained before. And these have looked like S4 or state space models. Many folks maybe have heard of Mamba as a recent model as well. And then our work today is focused on the Hyena architecture. So we'll be diving into that. Now, one of the things that we observed when we first started working in the space of long context foundation models or language models is that a lot of companies were really putting out these targets or new models with longer context, but all focused typically on language or computer code. And not much has been focused on in the past on long context foundation models for biology, which was interesting to see because some of the longest sequences out there are in biology, right? So the human genome is about 3 billion nucleotides, but the models being applied to these tasks in genomics typically were around 2,000 to 4,000 tokens. So there was just this huge gap. We thought that we could potentially make an outsized impact if we applied some of the things we were working on. Now, what we were really driven by was the potential for these long context models in genomics to help with better understanding disease, potentially help with drug discovery or target discovery, but also this concept of personalized medicine we were really intrigued by. So if you can imagine having something like ChatGPT that could fit an entire genome in context as a prompt, for example, and then you can query that genome for potential sources of disease, predict drug reactions, but also ultimately guide treatment options based on your specific genetic code. Now, those are all pipe dreams, things that we hope to achieve. For this work, we're going to focus on discussing genomics, the application of the model to genomics, which is the study of all genetic material, including its structure, function, and evolution. A couple key challenges that we observed and we wanted to kind of bring up were, we mentioned context length as a gap or constraint. We want to contrast this with other domains like language and proteins, typically shorter context. But this is a concept of also resolution or tokenizing. So when you're designing foundation models or language models, one of the key design choices that you have to make is what type of tokenizer or what's the smallest resolution you want to represent your data as. So in language, you typically represent a token, right, the smallest unit at the word or subword level. In proteins, you have single character levels, but the sequences are really short. In genomics, you have this requirement of both, right? Both very long context and high resolution, right? At the character level or base pair level. In particular, right, these things known as SNPs, single nucleotide polymorphism, these single SNPs, right, can alter genes, traits, protein functions, and mean the difference between having a disease or not. Like in sickle cell anemia, just one base pair change, a specific location causes that disorder. Now, specific previous models, right? We're looking at largely a good number of language models that were applied to genomics, and we followed a lot of the training recipes that they did, which was pre-train on a corpus of DNA and fine-tune on downstream genomic tasks that you might care about. A lot of great work in this space: DNA-BERT, GenLSLM, Nucleotide Transformer, paving the way in terms of applying foundation models to genomics. But again, context lengths were constrained. Just a fraction of the human genome was captured in these context lengths. and tokenizers. They all use these tokenizers to aggregate DNA into meaningful units. So typically 6 to 9 nucleotides into a single token, losing that single nucleotide resolution, which is something that we wanted to capture. And some of these models were also very large, multi-billion parameter models, which has its pros and cons in terms of training expressivity in your model, but also out of reach for a lot of labs, right? For a lot of academic labs or smaller labs more broadly. And so we wanted to address, be able to have a tool accessible to not just the biggest companies out there. Taking a step back, we wanted to ask, what are these genomic foundation models even trying to learn? And what they're trying to learn are useful and general representations from DNA sequences implicitly. What might that mean? Let's break that down a little bit into how that is applied to biology, or to DNA in particular. And the idea is to treat DNA largely as a language, with the assumption that there's an underlying grammar or syntax. And we do know this, right? There's, given a sequence of DNA, we have these coding regions or genes that can code for proteins, right? So there is an underlying pattern, of course. Now, that's just a small fraction of the genome. The majority of the genome, you have non-coding regions, or some might call it junk DNA. But we now know that this, non-coding or non-junk DNA actually regulates gene expression. So it doesn't code for genes, but it actually decides and what amounts, when and where to make these genes. And this is hugely important, right? Because it can lead to cell differentiation or different disease states. And so this interaction between coding and non-coding regions is extremely important. And we treat it, and perhaps these language models in general treat this as a combinatorial problem of the interactions between the coding and non-coding regions. So what are these interactions? Diving a little bit more into this, let's give an example. Say we have a couple of genes that we are taking a look at. What needs to occur is a protein complex, RNA polymerase, needs to bind to specific sites in your genome to kickstart the transcription process and create proteins in turn. Now, whether that complex binds or not is influenced by a number of DNA motifs that regulate that binding. These can promote, enhance, or even repress that binding from occurring. And that's how you get that regulation. And so some of these motifs can be really far from these binding sites. And so being able to have a model that can capture these very, very long range interactions is where models start to have challenges in being able to model these kind of phenomena. Now, the models that are being applied to these challenges, to these tasks, are typically the transformer architecture, which has been wildly successful in a number of domains, right? Obviously language, but also vision, audio, and proteins, right? For protein structure prediction, for example. Now, as great as the transformer model, or how useful as it's been, it's powered by this attention mechanism, this algorithm known as attention. It does have its trade-offs. So attention has, broadly speaking, high expressivity in terms of its modeling capability, because the receptive field, or the range in which it gathers information for a single neuron, is global. It has global context for a given input. Now, it comes at a cost in terms of time complexity, which is a measure of efficiency for computation. It's how we measure how much compute it requires for calculating this algorithm. It takes an n-squared operation, and so that in turn, just generally speaking, it's a very expensive operation. We want to contrast this with convolutions, which is another common primitive or layer in neural networks. And it sort of has the opposite set of characteristics. Its expressivity is typically on the lower side. It has a smaller receptive field. So the window in which it gathers information is usually local, right? But the trade-off is that it's much faster. It's typically a linear operation. And so you kind of have this opposite effect. And so the question that we asked and wanted to pursue in this line of work for Hyena architecture and also long sequences, is there a way to get the best of both worlds? Both high expressivity and compute efficiency. And so to do that, we started off with the original Hyena architecture, which we'll jump into, but we distilled two key properties of attention that we thought made it successful. And the idea is to try to reverse engineer that with more efficient primitives to try to mimic the attention algorithm. So the first property of attention that we want to mimic is this, or to have, is global context. And so what I have here is these squares that are representing tokens, right? Like words in a sentence, essentially. And the idea is that every token in attention has the ability to compare itself with every other token in the sequence. So it can be very far away, but it can compare itself. And this is what allows attention to be so successful at modeling all the interactions in its data. It's essentially doing this pairwise comparison, but doing it n number of times, and this is how you get the n squared operation. And the second property that we think is very important, that's less talked about perhaps, is this data dependency property. And so you have this attention equation here, but besides diving into the details of the equation, I want to sort of talk about the intuition about what the algorithm is trying to do. So the idea is to take these input projections and have the operation, the attention operation, dependent on the input itself. And that's what will allow it to adapt to new inputs, essentially, on the fly and make it so successful. I want to give a concrete example of what we mean by this adaptation. So we have this example in translation for in language of, say, trying to translate a German sentence to an English sentence. Initially you start off with the German sentence and a start of sentence token, just the beginning of your translation. And the idea is to auto-regressively, right, generate one token at a time in the English to translate this German sentence. Now, initially, what you're trying to do, you're essentially in attention. You're just taking you're learning to do a weighted a weighted sum of this German words to come up with the next English word one at a time. And so simply, you take this weighted distribution, you use that to create the next token, and then you feed that back in, actually. Now, this is your new context, what you've decoded so far, back as input. And so this creates a new distribution over the German tokens. And so you do this back and forth, essentially. And this is what allows you to autoregressively translate. And this is what we kind of try to picture as, the context changing, and then therefore the operation itself is going to learn how to change as well with the context. That might be a little bit hard to still kind of tease apart. It might be easier to see what we mean by data dependency by looking at an operation that is not data dependent. So if you have an image, so represented by these pixels, you have these kernels, a convolutional kernel that's used in CNNs that slides over the image left to right, top down, extracting features, right? And the idea is that this kernel that's sliding across, the kernel itself is not changing as you slide it, as the context is changing. The kernel itself is not changing. And therefore, that's what we want to contrast as a data-dependent operation. The kernel, the operator, is not changing with the input. And that's what we want to get around. But we wanted to modify this to enable that similar property that attention has. Okay, so kind of talked about that for a bit. So armed with those two properties, right, global context and this data dependency, now we can design a Hyena operator to mimic those properties. Our starting point is the transformer block, which is a block with a couple key layers. The idea is that you just stack these transformer blocks, consecutively, and voila, you have a transformer, right? Zooming into just a single transformer block, there's the attention layer, right? And then there's the MLP layer, the multi-layer perceptron. And these are the two key layers with a couple residual connections, but primarily these two key layers, right? We're going to keep the transformer block, but swap out the attention layer for what we call a Hyena operator. We're going to keep the rest of the transformer block in place. Now, if you were to walk away with, understanding what the Hyena operator is, it's made up of two key operations, right? We have the long convolution to capture that property of global context from attention, and the element-wise gate to capture the data dependency. So it's these two key properties, or these two key primitives that we're putting inside the Hyena operator. Now, zooming into this Hyena operator, how does data actually flow through the layer, right? So we'll talk about that next. The first step is to take your input X and pass it through and create these things known as projections, very similar to projections like attention, which we also use the same notation, but we use them differently, which are the key, query, and value. Typically, attention just uses the dense layer, like a dense layer in your standard PyTorch, but we also use a short convolutional layer on top of that as well. So key distinction there. You take two of those projections and you do an element-wise gate. That's literally just multiplying per element, multiplying them together to get the product. These input projections have the same shape, and so you can just multiply them together. And this is where we get that data dependency operation. We take that output and pass it through a long convolution. So long convolution, I haven't described that yet, but you can think of it for now as just a convolution normally done with a kernel sliding over the input, but the kernel itself is the size of the input. So it's a global kernel, and it's very long range. Typically, you have a short range, and we're making a long range run. And then finally, it passes through one more gate. And in large part, that is the Hyena operator. You have this option to kind of stack these or iterate between convolution and gates, but that's optional. The more important thing is to dive into what's inside this long convolution, like how to make this work, what makes it so hard, or what's been challenging about it in the past, and what's our value add in terms of the model architecture. So what makes it challenging is typically in the past, when folks have tried to use convolutions, they've seen a limit on how long you can make the kernel, right? Typically, when you have a short range kernel, you share a lot of parameters, and so it's much more efficient in terms of learning. But for us, if we're going to tie it to the input length, that means the longer your context you want to capture, the more parameters you're going to need. And that's where it makes it kind of challenging to be able to use long convolutional kernels. Second, right, you're still going to have this computational slowdown as well if you're going to make the kernel very long. So what we do in our work is to use this type of convolution that's known as an implicit convolution, or implicit long convolution in this case, to make the parameter efficiency better. And so we want to contrast this with what we're calling explicit or regular convolutions, where if you have an input that is 1 million length long, let's say, in 1D, the kernel that you need is also going to be 1 million. So it's going to be a large number of parameters. So the bigger the input, the more parameters. In implicit convolutions, no matter how long a given input is, you're going to use the same amount of parameters to learn that kernel, which is, in other words, using a fixed parameter budget to handle an arbitrarily long kernel. Now, how do you do that? We actually have a separate neural network, a mini neural network, really just an MLP, multi-layer perceptron, made up of a few dense layers, activation functions, that will actually learn to output the kernel weights. So this is what we mean by implicit. You're passing information through this filter function that will actually create the filter for you with a neural network. And so specifically, let's say you have input of length n, in this case 1,000. You take the positions of the index positions of the kernel that you want, so 1 through 1,000, pass that through the MLP, and it will output the weights of the kernel. It's not the final output yet. You can then take this kernel and then apply it and convolve it over the input to get the output, but they have this sort of intermediate step, which kind of alludes to the implicit convolution part. So, what's the intuition behind this? Why have this double step? That's somewhat convoluted. We have this analogy that might help folks to, just get a simple handle on this. Let's say, for example, you wanted to define a very long line on a 2D plane, right? One way to do that is to explicitly define every coordinate along that line, right? You'd have to do that very manually, and you could do it. You can define the whole line, but with defining every coordinate. But a simpler way, right, is just to learn a couple parameters, which is the slope and the intercept. And with those two parameters, you can define an arbitrarily long line. And so in this analogy, the long line is the kernel that we want, and the parameters that you need to learn, in our case for Hyena, is a neural network that will learn to parameterize that very long kernel. So hopefully that gives folks some intuition about why use implicit convolutions. Okay, so one more complexity about the long convolution. If you notice that when you have a long convolution, the time complexity, the compute required, still is actually very expensive. You have this n squared operation again. So it's kind of taking you back to square one with attention. It's just as computationally intensive at this point. So what we take advantage of to speed this up is to compute the convolution through the frequency domain. So for folks familiar with signals processing, if there are folks here, there's a convolutional theorem that states that a space convolution or normal convolution done in the time domain, just the way you normally think of the convolution, is equivalent to multiplication in the frequency domain. And so what does that tell us? It tells us that if we compute our convolution through the frequency domain, we can get a speed up from an n squared operation. So, this quadratic operation to an n log n. And so there's this point, this crossover point for very long kernels, which in our cases they're very long. And it becomes much more efficient to compute this convolution through the Fourier domain using the fast Fourier transform, the FFT algorithm. I won't dive into that here, but there's a long line of signal processing work that has helped us here, and we take advantage of. So another nice little intuition about what the Hyena model is doing. Essentially, with this convolutional theorem, you can think of the layer as basically doing convolutions in time and frequency, just kind of alternating back and forth. That's just literally just doing that back and forth. Okay, so now we have this new architecture that can handle longer sequences and compute it much faster. Now we want to take a look at the experiments and see, does it actually learn on DNA? Now the first step in creating foundation models is to train an unsupervised task. In our case, we're going to predict next token prediction. And so our starting point is the data set, which we use a human reference genome of just about 3 billion base pairs. We're going to sample a given sequence at a specified length, and then mask out the very last token of the sequence and simply just predict what's that last token, right? Just that simple task. But we're going to do that iteratively over many millions of batches, essentially, right? And then presumably, your model is going to learn the distribution of your data, right? All the interactions in the samples to be able to predict that next token better, essentially. And this is sort of the formulation for virtually all large language models or foundation models. And then we can take the trained model, the pre-trained model, and fine tune it on downstream tasks that we actually care about, right? So some of these genomic tasks include regulatory element prediction, chromatin profiles, or even classifications of species that we'll give some flavor about as well. So for the HyenaDNA model, we didn't just train a single foundation model. We actually trained a number of models where we varied the models by length, by depth, so the number of layers, the width, right, the model dimension, internal model dimension, and then also the context length to see how these models, how they learn or train differently given these parameters. So one of the observations that we observed, that we saw, is that with longer context, so as you increase the context ability of these models, we can achieve better performance in measured in perplexity, which is a measure that these language models use to essentially quantify how well you're predicting that next token, right, given that sequence, so that pre-training task. And so in this case, a lower perplexity is a better score. And so what we see here is that we vary different lengths of the context length, but also model size. And we observed that if you create a large enough model, you can take advantage of this longer sequence, essentially. There is a trade-off where if your model is not deep enough, not big enough, the longer context actually hurts. But we talk more about that in the paper. We also measure runtime. So, just purely looking at performance in terms of computational speed of a forward and backward pass through the model. And we compare with transformers. That's sort of the bar in terms of speed. And we see here is that for short sequences, Hyena is actually a little bit slower. But as you get to very long lengths, the separation in speed starts to grow quite large. We're in log space here, so log sequence length and log time. So it doesn't look as dramatic, but the separation is actually much larger. So at a million length context, which is about 500 times longer than previous foundation models, we're about 106 times faster. So it's a lot of X faster or longer, but essentially just to say that the quadratic computation of attention starts to really hurt. But for the HyenaDNA model, we scale much better, essentially. Okay, so lots of performance talk and pre-training talk. I'm going to start talking about some of the downstreams, or some of the actual genomic tasks that we started applying the model to. And in large part, a lot of them are these sequence classification tasks. So given a sequence, predict a single label for that sequence, for that input. And so in this case, we use this benchmark called GenomicBenchmarks, somewhat newer data set and somewhat simple also, predicting things like the presence of enhancers or different species or other promoters as well. And it's a binary task. And we do use standard fine-tuning. So we take a pre-trained model and then we fine-tune it on these tasks with a small training set, and then predict whether these sequences have these labels essentially. And we see that for HyenaDNA, there's a fairly large gap in some of these tasks, and this started to show signs that the model is actually learning something. We're not quite sure yet, but we want to get some indication. This was our first data set. A more difficult set of tasks was from the Nucleotide Transformer data set. And this set of tasks, there's about 17 or 18 different data sets. We're also including enhancers and promoters, but also things like histone marks and splice sites. And in this case, we're competitive on a number of the data sets, some by wider margins than others. But the thing that kind of got us excited was the difference in model size, right? So the HyenaDNA model was just about 1.5 million parameters versus a nucleotide transformer from this paper on the scale of about 2.5 billion parameters. So just orders of magnitude difference in terms of size, but also in terms of the pre-training data set. So they would use over 3,000 genomes in their data set, and we were just pre-training on a single human genome, which we were very excited about. I'll skip that. And in terms of we keep coming back to performance about compute. We also want to compare, just for that nucleotide transformer model to reach the same, that competitive level of performance. They trained on about 128 GPUs for a month, where we just trained for a single GPU for about an hour and a half. So just vastly different terms of resources required to reach similar level of performance. Now, so far what we've talked about were these somewhat short range tasks for these sequences. Now we wanted to talk about some of the longer range tasks. And the first one we designed was what we're calling, long range species classification. So the idea here is that, species can share a lot of similarity in terms of their DNA. And so to be able to distinguish between them, it could be beneficial to have longer context, right? To capture those differences. If you have a very short range model, it's going to be more difficult to tease apart those differences in DNA. And so that's what we observe. So we had this experiment where we gradually increase the context length and see how well it performs on this species classification for a 5-way prediction task for 5 different species. And so you have this trajectory that, longer context, better performance. You see the same thing happen with transformers, right? You have this upward trajectory, but it's at a point it becomes intractable computationally to actually use transformers because of that quadratic scaling inattention. Another type of experiment that we wanted to give a flavor for in this talk was, instead of standard fine tuning, what happens if you take some of the more modern techniques used in language models currently? There's this concept of soft prompt tokens, where you freeze the pre-trained model, right? You don't fine tune the whole model. You freeze the pre-trained model, and then you add loadable tokens in the sequence itself with the input. So you're just kind of stacking these weights into the sequence and feed that into the model, and then basically try to predict some kind of task like regulatory sequence function. In this case, we use the GenomicBenchmarks task again. And in this case, this is a nice way to allow your model to essentially be guided without actually changing the model itself, but guide the output with these soft prompt tokens and be able to still fine tune on downstream tasks. And what we saw is as you added more of these soft prompt tokens, the performance improves, right, up to a point, about 32,000 tokens. So we thought this was perhaps an exciting line of research that we wanted to showcase a little example about how you might be able to use these models differently. We also, toward the end here, we also take a look at the pre-trained embeddings to see, are these embeddings actually learning something in terms of structure of the data? And so we feed in sequences here of known labels, of different types of genes, different types of RNA, to see if there's some kind of class separation between the features, between the embedding types. And then we can do like a downstream task to basically predict if the model can learn to separate or classify these embeddings. And indeed, we can see better separation or better performance in this classification of a task between other models as well. Models that were just also, again, much larger, but shorter context. So I talked about what we've done so far in the HyenaDNA work. I do want to give a flavor about some of the things that we're excited about going forward. So we've kind of bucketized them in a couple of different silos. So we think in general that the model will work well in discriminative tasks, right? So things making a prediction, single class label for things like gene expression or splice detection, histone modification. And there's some folks pursuing that we've been talking to. But we're also really excited about the generative side as well. So can we actually generate sequences to some kind of controllable manner? And I'll talk about a couple as well here. So, we're borrowing some inspiration here from a recent paper that came out last week on DNA diffusion. So in that task, they essentially were able to map natural language prompts to sequence generation. So they had prompts like, generating a sequence that will activate a gene to its max expression level in cell type X, or a sequence that activates a gene in liver and heart, but not in brain. So some cell specificity. And the idea is that the model is going to generate the sequence following that prompt, right, in natural language. We're also, really excited about prompting not just on natural language, but prompting on gene expression profiles, right? If you can prompt on expression profile and then generate the sequences that would cause that expression profile, that would be very powerful, exciting application, we think. There's this other task that we thought was very intriguing as well, which we're calling miniaturization. So imagine if you had a sequence of some desired function, right? That's in the megabase scale. Is there a way to sort of shrink or miniaturize that sequence into the kilobase scale, but then still have that similar functionality, right? What we thought was intriguing about this was that in natural language, this task is a very common task. It's essentially just summarization of some text, right? And so we think this task would work well in biology or in DNA. Now the challenge is not necessarily this, sequence to sequence mapping task, but it's more so in the data set. Does the data set exist? Can we create it? Would it be too expensive to create? But it's something that we think would be very promising. And the idea is that you can potentially use this for gene therapy, right? If you have a limit on your cargo size that you can inject in terms of genetic code, if you can miniaturize that into a smaller payload, that would be very useful. And so with that, yeah, I just want to summarize very quickly. HyenaDNA, a genomic foundation model with long range and high resolution that is shown to be promising on a number of downstream tasks. But what we really want to do is get this in the hands of researchers to test this out on more realistic, more useful tasks. We've made the code base entirely public, and also the weights as well on Hugging Face. And there's a little bit of a community in terms of Discord to help try to stir up ideas as best we can. And very excited to see if folks have feedback and exciting ideas to apply it to. And so with that, thanks for listening, everyone, and I'm happy to take any questions if you have any.